My name is Irene Kaplow. I am an assistant professor in the departments of biological sciences and computational biology at Carnegie Mellon University. And today I'm going to tell you about my work, which involves relating enhancer genetic variation across mammals to complex traits using machine learning. Our world has an amazing diversity of animals with an incredible diversity of traits. And many of these traits have evolved multiple times during the course of mammalian evolution, suggesting that there might be some kind of shared genetic mechanism underlying the evolution of these traits. Yet the genetic mechanism underlying the evolution of most traits in animals are largely unknown. I previously focused on the trait of large brains relative to body size, which has evolved multiple times in mammalian history. So humans have much larger brains than cats and rats, and even than our closest relatives, the chimpanzee. Yet dolphins, which are much further from us than chimpanzees, also have much larger brains than we would expect based on the size of their body. And while my passion is understanding how traits have evolved, this understanding could also lead us to understanding disease mechanisms of human disorders related to those traits, such as microcephaly, which is being born with a brain that's too small. The way that we can study brain size and understand this genetic mechanism through which it has evolved is by looking at DNA sequences in different species. So our DNA consists of 4 nucleotides, A, T, G, and C, and within the DNA, there are regions called genes. And these genes are used to make RNA through a process called transcription. The RNA is then used to make protein through a process called translation. Proteins control the cellular processes in our cell, and these cellular processes could enable various traits such as brain size. So the first thing we thought to do is say, are there differences in actual gene sequences between mammals that might help explain the evolution of brain size? And a colleague of mine, Morgan Worthland, did this investigation, and she was not able to find any genes associated with the evolution of this trait. And what that suggests is that there are regions outside of genes that might be affecting the evolution of this trait. So the way that this can work is that there are regions in the DNA called enhancers. And what these enhancers do is they control in which tissues a gene is actually used to make RNA. So let's imagine in this toy example that we have a human enhancer that is involved in activating a gene and that has an A at a certain position. So this then enhancer is responsible, let's say, for allowing this gene to be transcribed in the brain. Now in this toy example, let's imagine that our closest relative, the chimpanzee, has a G at this position of the enhancer. Well, that G could then mean that the gene is no longer able to make RNA in the brain, which could then mean that the protein corresponding to this gene is not made in the brain, which could affect brain cellular processes and in turn affect brain size. However, finding enhancers associated with the evolution of a trait is difficult, and this is true for a couple of reasons. First, most mammalian enhancers have tissue-specific activity, since their job is to control the tissues in which a gene is used to make RNA. But the DNA sequence in every tissue is the same. So if we get DNA from, say, a blood or skin sample, which doesn't require sacrificing an animal, we're not going to know where the enhancers are in a tissue like the brain that might be more relevant to our trait. Second, enhancer function can be conserved even when nucleotide level conservation is low. So I want to illustrate what I mean by this through an example that came out a few years ago. And this is an enhancer in humans whose function is conserved in zebrafish and even all the way out to sponges. However, if you align the A's, T's, G's, and C's in these nucleotide by nucleotide, you don't see a great alignment. And what's happening is that there are various sequence patterns within the enhancer that are preserved. Those are indicated by the colored boxes. And the way many enhancers work is that there are actually multiple sequence pattern orders that can allow for the same function. So we have these different orders in different species, which means the enhancer is still functional because all the patterns are there. But if we align it nucleotide by nucleotide, we see low conservation. However, what this does suggest is that sequence feature conservation can be indicative of tissue-specific enhancer activity conservation. So the idea that we came up with is to use machine learning models to predict enhancer activity across species. And the way that this works is something that we can get through doing experiments is a matrix that looks like the one on the top. Each row is an enhancer, Each column is a mammal, and what we can get is enhancer activity data from a small number of species where we can actually access brain samples. We can then do is take our sequences underlying enhancers and non-enhancers and use them to train a machine learning model to predict enhancer activity in the brain. We can then take the rest of our species where we have a DNA sequence that we were able to get from something like blood or skin and predict where in the DNA our brain enhancers are, allowing us to fill in most of this matrix. So more specifically, to study the evolution of brain size, what we did is we collected brain enhancer activity data from a few species, and specifically we used the motor cortex region of the brain. We then took the sequences underlying our enhancers and non-enhancers and trained a machine learning model to predict brain enhancer activity. What I realized while doing this is that the evaluation criteria for these kinds of machine learning models were not sufficient for actually evaluating the task we care about, which is predicting enhancer activity differences between species. So I created a new set of evaluation criteria and compared both my method to some previous published methods, which you can learn about in my paper in BMC Genomics. We can then make predictions of brain enhancer activity at regions corresponding to our brain enhancers across large numbers of species. We can associate our predictions with brain size and evaluate the statistical significance of our association. So using this method, we found a few dozen enhancers associated with the evolution of brain size, and I'm just going to show you one example. Here is an enhancer in the red box, and you can see that it's near the gene SAL3. Now, this enhancer has our predictions of enhancer activity in the brain have a strong correlation with brain size relative to body size in both the whales or the cetaceans and the great apes. And these are the two clades of mammals that have the largest variation in brain size relative to body size. So I then wanted to know, well, what does the gene SAL3 do? So my fantastic former undergraduate mentee, Daniel Schaefer, did a literature review on SAL3. And what he found is that SAL3 is expressed in the human cerebral cortex, and it regulates the maturation of neurons. So I then asked the question, this enhancer is about 1 million bases from SAL3. If we look at three-dimensional interaction data in the human brain, does it have an interaction with the gene SAL3? And what we found is that in the adult cortex, this enhancer does have a significant interaction with SAL3. You can learn more about these and our other results in my paper that was published recently in Science. So in the future, my lab is going to be extending this work to study how metabolic traits have evolved in mammals. And we're going to be focusing on initially on two tissues, the liver and adipose tissue. And some traits we're really excited about exploring are diet traits like carnivory and herbivory and hibernation traits. There are also a number of computational challenges we want to overcome to be able to broaden this work, both to traits that differ between very closely related species and to traits that differ between much more distantly related species than just the mammals in our previous study. So for closely related species, what we often see is not that an enhancer gains or loses activity, but rather that its activity increases or decreases. So to train machine learning models for this task, we're going to need to train not just classification models, but also regression models. And while many have trained regression models for genomics tasks before, the current evaluation criteria don't directly address our task. So we're going to need to develop new criteria and then figure out what the best model training approach is. We also need to be able to study more distantly related species. We're currently assuming that we have the same enhancer affecting our trait across all of our species, but once we have more distantly related species, what we're probably going to have is situations we have different enhancers for different groups of species all regulating the same gene. So we're going to need to come up with a principled way to account for that situation. And once we can do that, some things I'm excited about are studying more distantly related mammals than we've previously studied before. This is an example of one that we weren't able to study before called the tenrec, which is very interesting, mild form of hibernation. Also studying birds. There's now sequences of 600 mammals of birds that we can leverage. And finally, studying even more distantly related vertebrates. My lab is going to be getting involved in the Vertebrate Genomes Project, which is in the process of sequencing hundreds of vertebrate genomes. If you want to learn more about this, feel free to check out my website. Thank you very much.